Welcome to my channel Adhyan with Flow. Our new topic is generation and conduction of nerve impulse. Before starting this explanation, I want to clear some terms like what is the channels, what is the pump. Alright guys, I want to clear something new. We all know that every cell covered with their cell membrane. We call it as a cell membrane, plasma membrane. Alright. So this plasma membrane have special feature like this plasma membrane is selectively permeable selectively permeable for different ions. Alright guys, this plasma membrane is selectively permeable for ions, different ions. It may be molecules, it may be proteins, and it may be compound. Okay, so if someone want to cross this membrane, any molecule, any molecules, either ions, either compound, who want to cross this membrane, they need permission. They need permission to cross the membrane. Who give them to permission? Yes, the name is channels, the name is transporter, the name is carrier, the name is pump. Okay, these are protein molecules. These are protein molecules. They are work as a carrier. They work as a carrier to transport molecule from outside to inside the cell and inside to outside of the cell, both inside and outside. They do their work to transport any kind of molecule. Some channels and transporter are required ATP, and someone cross the molecules and ion free of cost without ATP. So channels is ATP dependent. It does not need any ATP for transporting the molecules. And pump, like pump, they are ATP. Wait a minute. They are independent and they are ATP dependent. If any molecule combined with pumps, so first of all, they give them ATP. Then these pumps transport the molecules. First, give some ATP, and then they transport their molecules. So, I hope you can understand this term that plasma membrane is a hydrophobic. No one can pass the plasma membrane outside to inside and inside to outside. But there are some carrier proteins. There are some mediators protein over there. They can transport any molecule and ions, proteins, compound to outside to inside of the cell and inside to out of the site. Okay guys, this term is clear. What is channel and what is pump is clear. I think very clear. Now another important, this is the plasma membrane of axon, exolema. Alright guys, this is the plasma membrane of axon. The special features of axon layer is they contain outside positive charge, like they have excess amount of sodium ion, and inside the cell they contain negatively charged ions like potassium, like potassium. Clear this term? Is it clear guys? Let me show you. Here, look it here. This is the axon. This is the axon. Inside the axon they contain negative charge. They contain negative charge, and outside of the cell they contain positive charge. Means outside the cell sodium ion is more and more and more, and inside the cell K plus ion more in number. So I repeat, inside the cell K plus ions more, so negative charge more, and outside the cell membrane, outside the plasma membrane, positive charge more, so membrane become positively charged because sodium ion exists in outside of the plasma membrane. Clear this term? Inside the cell there are more and more negative ions, and outside the cell positive molecules, ions over there. Clear guys? This is inside and this is outside. Let's move further. And now let's start generation and conduction of nerve impulse. We know that dendrites we know that dendrites take signal from outside. We know this thing, that signals received by dendrites, then dendrites transmit their signal towards cell body, and cell body transmit their signals towards axon. We all know this thing. What is the way of this signal transition? This signal transition, what is the mode of transition? We know that signal attach at dendrites, because dendrites plasma membrane have receptor and signal molecule ligands can bind with them. Okay, we all know that signal come and bind with dendrites. Then what kind of signal reach towards cell body and cell body towards axon? What kind of signals? What kind of transduction is over there? It's electrostatic. There is fluctuation in positive ions and negative ions. Look at here. I'll explain in detail. When this is the axon, assume this is the axon, long cylindrical structure. This is the axon. This is the axon part. Here, what's the changes over there? This is the cell body, assume it, guys. This is dendrites. Okay. Here, pumps are located. Both the side. Pumps are located. The name of the pumps are sodium potassium pumps. Sodium potassium pumps. Alright. This structure of plasma membrane, outside the positive ions and inside the negative ions, this cell is present in their polarized form. Polarized form. When stimulus is not present, stimulus is not present, when this is the position of every ions. But now if we get stimulus from surrounding, then what happen next with exolema? Oh wow, look at here, look at here. If we get stimulus, if we get stimulus from surrounding, then what happen next? These are pumps, these are pumps. Look at here, these are pumps. The name of the pumps are sodium potassium pump. I'm writing here sodium potassium pump. And I told you before that pump work in ATP dependent manner. They need ATP to transport the ions. Okay, this is axon part. This is the long axon part. Clear, guys? This is the axon part. Now, if we get stimulus, then polarized form change. Polarized form changed and cell convert into depolarized form. How? We know that outside the cell positive ions are present and inside the cell negative ions are present. We know this thing. Then when we get stimulus, the positive ions enter by pump in inside and negative ions out by sodium potassium pump. Understand? So this positive ion enter inside, look at here, enter inside the cell body. Here also this positive ion enter inside, and here negative ion outside, here negative ion outside the cell, negative ion outside the cell. Vice versa, na? Positive ion in and negative ion out, negative ion out of the cell and positive ion enter in cell. When we get stimulus, and this process carry forward all type, plus in minus out, plus in minus out, plus in minus out. This is transduction way. This is the transduction way, guys. Now how then cell membrane become normal? How? First of all, when, wait a minute, when signal move forward, when signal move forward, then previous position become normal. When signal move forward, when signal move forward, then this position become normal. How? Again this negative ion come inside and positive ion move inside the out. Okay, now this is the fluctuation, this is the depolarization state. When cell want to come in their normal stage, then what will happen? Then what will happen? Signal goes forward, then previous step is become normal. Look at here, positive charge out and negative charge inside, positive charge out and negative charge inside. Then signal move forward, then positive charge again out of the cell and negative charge who goes outside, who come inside. Okay, now this is the structure of polarized form. I made it here depolarized form structure, depolarized structure. This is the plasma membrane of axon. Now this is the pumps, and this is the depolarized form, that minus charge come outside and positive charge go inside, minus charge come out and positive charge inside. This is the depolarized form, and we want that cell become normal, cell come in their normal form, then this process is repolarized. What happens then? Again after depolarization, cell come into their real normal stage. This is the cell membrane This is the cell membrane. This is the pumps. Now pumps again transfer positive ion outside the cell and negative ion inside the cell. Positive ion move outside the cell and negative sign come inside the cell. Okay, this is the polarization form, depolarization form, and repolarized form. I think this is term clear. Whatever I explain, all things are written here. Whatever I explain, all things are written here. Resting potential means positive charge outside and negative charge inside. Action potential means negative charge outside and positive charge inside. I'll made for you, this is the plasma membrane. In resting potential, positive charge outside and negative charged molecules inside. In action potential, this is the plasma membrane. The positive charge goes inside and negative charge goes outside the cell membrane. This membrane potential, resting potential, maintain in the absence of stimulus. And this condition potential present in the presence of stimulus. Now they have their pumps over there. The pumps are situated here. Like I drew a pump. This is the pump. This is the pump. Okay. And this is the binding sites. Here potassium ion can bind. Here potassium ion can bind. And here sodium ion bind. Here sodium ion bind. There are 3 site for sodium and here 2 site for potassium. So at a single time 3 positive charge go inside if cell membrane is in their action potential. So sodium ion bind outside medium and bind with their sides and come inside cell, and more potassium inside the cell, they bind sodium potassium pump and they go outside the cell during action potential. Okay, this is the sodium potassium pump. Okay, so this is all about generation and conduction of nerve impulse. Whatever I said, this is written here. 3 positive outwards and positive potassium ion inwards. When cell repolarize again, and this condition also found in when cell in their polarized form. Okay, this is the structure of polarized membrane. This is the structure of depolarized membrane. This is the structure of repolarized membrane. This is the structure of resting potential. This is the structure of action potential. This is all about signal transduction. Now next thing, transmission of impulse, we will cover in our next video. So if you like my video, then please subscribe my channel and support me. Share my channel. Thank you for listening me. Thanks a lot.